Structural elucidation spectroscopy, miscellaneous
Okay. What are the types of electronic spectroscopy?
absorption spectroscopy, emission spectroscopy, ma'am.
Mm.
In that, example of molecular level, UV and visible, ma'am.
Mm. Mm.
And atomic level spectroscopy, example atomic absorption.
Mm. Okay, about structural elucidation.
Infrared, ma'am. Preliminary
Mm.
test of infrared, NMR, nuclear magnetic resonance, ma'am.
Mm.
Electron spin resonance,
Okay.
uh, X-ray diffraction, mass spectrometry, ma'am.
Okay, ma'am. Miscellaneous.
Miscellaneous, under this polarimetry,
Mm.
thermal analysis.
Thermal analysis. Okay, good. Thank you. Anybody else? From boys? Anyone? Make it fast. One of you explain. Subadarshini. Subadarshini, are you explaining?
Yes, ma'am.
You can start now.
Yes, ma'am. What, ma'am?
Say about spectroscopic analysis, what we have discussed in the last class.
Electronic spectroscopy and emission spectroscopy. Under spectroscopy, electronic spectroscopy, structural elucidation and miscellaneous. And under that, electronic, it is divided into two, that is absorption and emission spectroscopy. In absorption, molecular level is UV visible, and atomic level spectroscopy is atomic absorption, and in emission, molecular level is fluorimetry, and in atomic level it is flame emission. Under structural elucidation, it comes under infrared, nuclear magnetic resonance, electron spin resonance, X-ray diffraction, and mass spectrometry.
Okay, ma'am. Miscellaneous. What are the miscellaneous techniques?
Polarimetry and thermal analysis.
Okay, ma'am.
Thermal
Good.
analysis.
Okay, good. Thank you. Anybody else from boys? You can mute your mic, huh? Subadarshini. From boys side, Jackson.
Yes,
Jackson.
ma'am.
Uh, say about the types of spectroscopy.
Absorption spectroscopy, emission spectroscopy, ma'am.
Hmm.
I mean, absorption, invisible and atomic absorption spectroscopy, and in emission, fluorimetry, tree and flame emission spectroscopy, ma'am.
Okay. About structural elucidation, name the structural elucidation spectroscopy.
In infrared, nuclear magnetic resonance, electron spin resonance, mass spectrometry, and X-ray diffraction.
Okay, good. Miscellaneous techniques.
Two types of analysis, ma'am. Polarimetry and thermal analysis, ma'am.
Okay, ma'am. Very good. Thank you. Eca Qa, Subhadashni and Jackson.
Thank you.
Thank you all. Let's move on to the remaining part of introduction now. It is about electrochemical methods of analysis. This is the third part of quality control methods as per your syllabus. Okay. In that there are three types of electrochemical methods of analysis we are going to study. The first one is potentiometry, then conductometry, then polarography or amperometry. Okay. What type of chemicals generally can be analyzed by electrochemical methods means mostly ionizable substances. In solution form, mostly all the analyte will exist in solution form as its ions. Okay, so how much concentration of ions are present and what type of ion is present? Both qualitative and quantitative analysis on ions are carried out through electrochemical methods of analysis. Since ions will carry charge so that they can conduct electricity, so based on that concept we'll be analyzing your sample through electrochemical methods of analysis. Okay, in that potentiometry and conductometric principles are more or less similar, where in potentiometry will be analyzing potential of the sample in the form of millivolt or electromotive force. What we'll be analyzing by potentiometry? Millivolt or electromotive force for the sample. That means blindly can say potential of the sample is analyzed. Okay, comparing with standard potential, sample potential will be analyzed. Wherein conductometry, conductivity of ions will be, ionic conductivity will be analyzed in the form of molar conductance. Okay, using conductivity cell will be analyzing will be analyzing samples by conductometric method. Why you need to study this electrochemical methods means how it is related to biological samples means all biological fluids will be composed of many biomolecules. Hope you have studied different types of biomolecules exist in the body fluids in medicinal biochemistry, right? So those biomolecules are existing in the body in solution form. What is the solvent present in our body? It is water. So it will be mixed in water as its ionic form. So to analyze the level of some biomolecule, either atomic or molecular form, it will be analyzed using potentiometry or conductometry or polarography. Based on their properties, will be moving on to selecting particular electrochemical methods of analysis. So this technique is commonly applicable for analyzing biological samples, whichever has ions in it, mostly electrolytes. This all can be analyzed by using potentiometry, conductometry and polarographic techniques. Okay, wherein polarography or amperometry will be analyzing polarizable substances. Polarizable means they will be oxidizable or reducible at electrode. We'll be using electrodes for analyzing it. So definitely at electrode surface, your sample will be either electroreducible or oxidizable. So those type of substances can be analyzed by polarography. Okay, amperometry also following the same principle, where diffusion current is measured. Diffusion current that is conducted by the sample, diffused from the sample, is analyzed by amperometry. Okay, so both are same only with the different acronyms, polarography and amperometry. So then nearly 4 techniques, including amperometry, 4 techniques you are going to learn under electrochemical methods, that is potentiometry, conductometry, polarography and amperometry. Okay. Next, about quality assurance. So quality control parts are of 3 categories. First one is chromatographic separation. Then second is spectroscopic characterization. Then third is electrochemical methods of analysis using electrodes, wherein spectroscopy will be using electromagnetic radiation for characterization. For separation by chromatography, we will be using two phases called stationary phase and mobile phase. So likewise, samples are analyzed through these quality control methods to identify. Our main aim of analysis is what? To identify the quality and purity of the drug substance to ensure its safety and efficacy. Okay. And how we will means get 100% quality for the drug means by following quality assurance procedures. There are 4 categories as per your syllabus. First one is guidelines. So these are all the organizations who have prescribed standard guidelines for manufacturing and all laboratory practices, good manufacturing practices, good laboratory practices, good clinical practices. So all the guidelines are prescribed by different organizations that is going to be learned by quality control, quality assurance procedures in that GLP, ISO 9000, TQM, ICH. These 4 guidelines we are going to learn. GLP is good laboratory practice. And ISO is International Standards of Organization, 9000 standards. Then total quality management, then ICH is International Conference on Harmonization. Apart from 4 standard guidelines, ICH is most important one since it prescribes all analytical principles to study all quality safety guidelines. Okay, based on our analytical parameters will be covered in ICH, International Conference on Harmonization is the committee. Okay. Committee prescribes all standard guidelines for quality control methods. Okay, ICH. And other practices are also adapted for getting 100% quality ensured drug. Okay. The next part is calibration. If before using instruments, either chromatographic or spectroscopic or any electrochemical instruments, we need to calibrate it. Calibration is done to ensure the getting accurate results. Okay. We can get 100% accurate result by means of calibrating analytical instruments. So what is the calibration? What are the different types of calibration? When we will be calibrating? What is the frequency of calibration? What are the instruments are calibrated? What time period it is calibrated? What is the calibration standard? This all will be discussed under calibration part. Then is validation. Equipment validation. It is what is difference between it is a interrelated calibration and validation. Validation is nothing but to get a consistent result. Whenever you do any experiment or process repeatedly, it has to give consistent result. It should not be always accuracy also important, but it should be repeatable. Okay, always you need to get accurate result. That is achieved by validation techniques. There are different types of validation: equipment validation, process validation, and method validation. For analysis, method validation is most important for developing any analytical method. Okay. And fourth part is regulatory aspects. What are the various regulatory bodies like patents, copyrights, for whatever drug you have manufactured or innovated, and that is registered as per regulatory bodies. Okay, whatever they have given the rules and regulations, accordingly we will be registering our innovation based on getting patents, copyrights, trademarks, whatever, all the things. You should have known, you should have come across many products that is having copyrights and trademarks for particular products and innovations, creations, whatever. Okay, so it is applicable for our pharmaceutical products also. So these are all the various aspects under quality assurance you are going to learn as per syllabus. Okay, it's about introduction on your syllabus. So what are all the techniques we have seen? Under separation technique, there are 2 types: planar and columnar. Under planar, paper PLC and HPDLC. This is part of quality control. Okay, in that first is separation, paper TLC and HPDLC for planar. For columnar, column adsorption, HPLC, ion exchange and GLC. For separation techniques, miscellaneous, electrophoresis, gel filtration, and affinity chromatography. Then the second part of quality control is spectroscopy, that absorption and emission, UV visible, atomic absorption, fluorimetry, and flame emission. Where atomic absorption and flame emission is atomic level analysis, where UV visible and fluorimetry is molecular level analysis. And for predicting a chemical structure, these are all the spectroscopic analysis are used: infrared, NMR, ESR, mass, and XRD. Then remaining techniques are polarimetry and thermal analysis, DSC and DTE. Don't confuse this polarimetry with polarography. Polarography is different. Polarimetry is different. Polarimetry is applicable for determining optically active compounds, okay, enantiomers, isomers. And stereoisomers will be analyzed by using polarimeter, where polarography is applicable. It is electrochemical method and it is spectroscopic method. It is the difference. And under electrochemical methods, potentiometry, conductometry, and polarography, where potentiometry, conductometry, this all analysis you will be performing in practicals also in the lab. Okay. It is related with your practicals. Theory, whatever you learn, you will be performing in practical and you will learn. You are going to use what type of instruments means under chromatography, you will be carrying in the lab paper, TLC, HPTLC, and column for project purpose you will be using. So you people will be means learning about HPLC. If you want to know about GC, we'll get demo from our university so that you can learn. And regarding electrophoresis, gel electrophoresis also available in our campus, so you can learn doing electrophoresis technique. And absorption, UV visible, fluorimetry, flame emission, these three techniques you are going to do in practical also. Where atomic absorption is not available, only UV visible, fluorimetry, and flame emission you will be carrying with the instruments during your practicals. And structural elucidation, we don't have any instruments mentioned here. Where IR is available in our university, so if you want to get demo, you can go there and learn. Where about other instruments, only available in industries, not in institution. Polarimeter is available, where DSC, DTA instruments also available in industry, not in institution. Where potentiometry, conductometry you will be analyzing in the lab itself. Okay. And these are quality assurance guidelines. Under quality assurance, you will perform calibration, okay, on instruments. Validation comes when you are going for research purpose project. You are carrying out in lab. Mostly you will be depending clinical work, so you will not be carrying out validation. Only you will be carrying calibration by using the instrumental methods. Okay. And thank you all for having an introduction on your means listening for your introduction on syllabus. In the next class, we will be discussing about introduction on chromatography. We'll be moving on to the first part of quality control method. And that we'll be seeing about introduction on chromatography, that is separation techniques. Okay. Is there any doubt? This introduction part. Let me share your
No, ma'am.
also. We'll have discussion on syllabus, how it is related with the presentation. Okay, it is not as such similar to the order given in the syllabus. I have changed your syllabus pattern according to the titles what I have divided in the PPT. Okay, what I have presented in the PPT. Accordingly, we'll be learning each and every technique. Can you see the syllabus copy now? Empa?
No, ma'am.
No. Wait. No. Okay. Okay. Hope you will be seeing now. So your theory,
Yes, ma'am.
thank you.
Yes, ma'am.
Your theory syllabus, it is your subject code 3.2 pharmaceutical analysis theory. Here you have 1, 2, 3 and 4 units. Okay, 4 categories of topics. In that, first you have given with the quality assurance. It is actually full theory oriented. If we discuss in the beginning, you will get bored. So it is that's why I have converted this part as a fourth part of the syllabus, quality assurance, and I have divided this chromatography, electrochemical methods, that is electrochemical analysis and spectroscopy in the beginning. In that, comparing electrochemical methods, spectroscopy is most important part, so that I have brought that after chromatography. Okay. So in chromatography, what are the things we are going to study? Column chromatography, PLC, PC, that is paper chromatography, ion exchange, HPLC, HPDLC, gas chromatography, electrophoresis, gel filtration, and affinity. This I have divided according to the planar, columnar, and miscellaneous. Okay, here planar is PC, DLC, and HPDLC. So concepts behind these techniques we will be discussing in the beginning. Then column in that column, gas, HPLC, gel filtration, affinity, this all will be discussing later. And then electrophoresis, lastly we'll be discussing. Okay, so according to the concept wise I have divided, so that with the continuation you can learn all the topics. And in practicals also, why I'm moving with planar chromatography first means you will be doing practical simultaneously in the lab, first chromatography only, so that if you learn the concept behind this planar chromatography, it is easy for you to follow in the practicals also. That's why. So after chromatographic analysis, we'll be seeing about spectroscopy. Here absorption. In absorption emission is included in the same category. Here somewhat vast syllabus, so, but we will be learning all the concepts with means comparison and difference between each and every spectroscopy will be learning, so that it is easy to understand all the concepts. By seeing syllabus, don't be afraid, it will be very easy. Concepts are very easy when we learn each and every topic. So absorption is UV visible and IR. Emission is fluorimetry and flame photometry. Here atomic absorption and atomic emission is given separately. This atomic emission and flame emission both are similar only, but here it is given separate topic. Okay. Then NMR and ESR, structural elucidation spectroscopy, mass, polarimetry, XRD, thermal analysis. In this DSC, DTA you are going to learn. These are all the topics you are going to learn. If we go one by one as per syllabus, it will be confusing, so that your syllabus is changed according to the concepts what we require. Okay, and electrometric methods, potentiometry, conductometry, polarography, and amperometric titration. These both are similar only, only variation with the type of analysis. Potentiometry, conductometry also different techniques. So there are 4 techniques you are going to learn under electrometric methods. That's all. And about your practicals, it is also code with your subject code is 3.2 practical. Here your practical also is divided as per the theory, quality control and quality assurance methods. And already the experiments are prescribed in the syllabus itself. So according to the syllabus content, you will be carrying all the experiments. Okay. First chromatographic techniques, then UV visible spectroscopic techniques, then titrations. This titrations you will be carrying based on the third part. According to the theory syllabus, it is third part now. So after analyzing all spectroscopic methods, you will be moving with titrations. Okay, these are all different spectroscopic techniques. Okay, once you receive the manual, you will be knowing how it is divided, all the techniques, how it is divided according to our means theory syllabus. Okay, you'll be learning and doing practical, so that you will get clear concept regarding all the instruments, how it is applied for the quality control purpose of drugs. Okay. Okay, thank you all. In the next class tomorrow, first hour, you will be having introduction on chromatographic techniques.
Thank you, ma'am.
Thank you all. We'll leave now. I'll
Thank
let
you,
you
ma'am.
mark the attendance. Okay, thank you.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Rafiq, Johnson, hello, Rafiq.